Bismillahirrahmanirrahim. In the name of the most merciful and the most beneficent. Today we will discuss about local and global alignment, one of the most important topic in bioinformatics, or you can say in uh, early lectures of bioinformatics. Depends on local and global alignment. Alignment, as you guys know, that aligning some sequences, one or two sequences, or more than two sequences. So first of all, we will have a look to the introduction of sequence alignment, as sequence alignment is a way of arranging the primary sequences of DNA and RNA or protein to identify different similar regions of protein, DNA, or RNA that may be consequence of functional structure that helps to understand the evolutionary function or evolutionary relationship between the two sequences. The known sequence is known as reference protein or reference sequence, and the other is as target or query sequence. So the sequence alignment is made between a known sequence and unknown sequence, or between two unknown sequences. The known sequence is known as reference sequence. However, unknown sequence is known as query sequence. So what is the main specific principle of alignment? As alignment can reveal homology between two sequences or the similarity between two sequences. Similarity is descriptive term that tells about the degree of match between the two sequences. Sequence similarity does not always imply on a common function. Doesn't mean that if the sequences are similar, the function will be similar. However, similar sequences has usually similar functions. So conserved function does not always imply similarity at the sequence level. However, it doesn't apply all the times. Usually similar sequences has similar structures, and similar structures has similar functions. Usually, convergent evolution sequences are highly similar, but are not homologous. On sequence alignment is the most important tasks in bioinformatics, as we already discussed. On sequence alignment, it helps for the prediction of unknown protein functions, database searching, gene finding, sequence divergence, sequence assembly. So here is some basic concept about sequence similarity. Homology. Genes that derive from a common ancestor gene are known as homologous genes, that are derived from a or duplicated from a one single ancestor gene. However, orthologous genes are homologous genes in different organisms, and paralogous genes are homologous genes in one organism that derive from a gene duplication. Homology mean one single gene just one ancestor gene just has two similar genes, or the genes derived specifically from one gene is homologous genes or homology. However, orthologous, that same gene present in different organisms. Suppose gene A is present in human, chimpanzee, mouse, rat. However, paralogous genes are homologous genes that present in one organism, however derived from gene duplication. These are known as paralogous genes. Suppose gene has different functions but present in same organisms, and this gene has same function but present in different organisms are orthologous, and gene with different functions present in same organisms are paralogous. So gene duplication, one gene is duplicated in multiple copies that therefore free to evolve and assume new functions is known as gene duplication. So you know about mutation. Mutations occur when there is any change or replace in any nucleotide, like ATA to ATA to AGA. Insertion of any new nucleotide between any existing nucleotides, like AA to AGA. For deletion, you know that the deletion of any specific nucleotide from one position, like ACTG to AC-G, and insertion and deletions collectively known as indels, or called as indels and insertion or a deletion. There are two main concepts for sequence alignment, that is global alignment and local alignment. As the word explains, in global alignment, alignment was from end to end. Suppose there are two genes, one gene A has 1 200 amino acids and gene B has 1 201 amino acids. From 1 to 100 amino acids alignment along with 1 to 101 amino acids or nucleotides are global alignment. However, local alignment, as you the word explains, that it is in patches for motif recognition. Suppose there is a gene has 1 to 100 nucleotide, and gene B has 100 to 110 nucleotides, and only the portion of 30 to 35 from gene 1 and 90 to 95 from gene 2 has similarity. Then local alignment will perform that, the locally check that which specific region has similarity. And the local and global alignment find the similarity between two DNA sequences by finding a good alignment between them. The biological problems of sequence alignment we have, like here you can have a look that it is a DNA sequence 1, this one, and then DNA sequence 2. Then there is an alignment. For this alignment, you can see T with T, C with C, and go on. However, there is here in DNA sequence 1, here we have a C. However, here we have a G. So it is a mismatch. It is not a match. However, C and C is a match. Then we again has a mismatch, and here we has also a mismatch. Then we have a gap, because here we have GGG and there we have a gap. If we will not give the gap, then this C will be this with this G, then A with this G, A with this G, this C with this C, then C with A. So whole alignment will be disturbed. So if there will be 3 gaps, this whole portion will be aligned of sequence DNA sequence 1 with the DNA sequence 2. So these gaps are added. So this is the basically concept of sequence alignment. Sequence alignment is an arrangement of 2 or more sequences highlighting their similarities. The sequences are padded with gaps. Here we have dashes and gaps, so that whenever possible or wherever possible, columns can contain identical characters from the sequences involved. Here in this example, we have this, we dissect this, that how, why we add these gaps, and these gaps just help to align the other part of the sequence of sequence DNA sequence 1 and DNA sequence 2. Types of alignment based on the completeness, it is classified into 3 different types known as global alignment, local alignment, and semi-global alignment. So we already discussed about global and local alignment. So here we have an example, like here in sequence 1 from 1 to 11. However, sequence 2 has only 6 nucleotides. However, for global alignment, we have to add gaps here, and we have to align the whole sequence, first sequence with the second sequence, that is the global alignment. However, in local alignment, like here, it just skip the nucleotide 1 and 2, and also 9, 10, and 11. However, this portion is totally is only aligned, and it showed the aligned alignment, that is local alignment. However, in semi-global alignment, because it is the major, it was the big portion that was not aligned, so he just skipped the big portion, and it takes the small portion, that is the semi-global alignment. Like it is not fully global and not fully local. However, it has the properties of global and local, both the alignments. So depends on the nature of experiment, that which type of experiment you are going to do and which type of alignment you need. So that's why you have to choose that specific alignment for your analysis. So for algorithms, needleman, wuntch, and Smith Waterman and BLAST, these 3 algorithms are studied and applied widely in bioinformatics. However, first needleman wuntch was discovered by needleman and wuntch, and that gives a pairwise global alignment only. How after that pairwise local alignment was produced, was generated, the algorithm generated by Smith Waterman. After that, BLAST. So all these algorithms has their own specificities and have their own characteristics for specific alignments. So pairwise sequence alignment methods, they are concerned with finding the best matching pairwise for local or global alignment of proteins or DNA. Pairwise mean one pair with the other pair, and it may be a local or global alignment. Typically, the purpose of this is to find homologous of the gene or gene product in database of known examples, that how these gene evolve, where duplications happen, which similarities it has. If we have a known gene and with the known function and with the known motif and with the known domain, So and we have an unknown protein, and then we have an alignment, good alignment with the known protein, then we can assume that the unknown protein has similar functions and similar properties. This information is useful for answering a variety of biological questions: the identification of sequences of unknown structures or functions, the study of molecular evolution, and many other. So in global alignment is in actual the matching of all the residues of two specific sequences for their whole length. It matches the identical sequences, as we have a look in the previous figure, that the sequence 1 has 1 to 11 nucleotides. However, sequence 2 has only 6 nucleotides. However, it aligned. That is the global alignment. To align every residue in every sequence are most useful when the sequence in the query set are similar and of roughly equal size. Then you can perform global alignment. If suppose gene A has 1,000 nucleotides and gene B has 1,020 nucleotides and 1,000 or 50 nucleotides, then you can jump on global alignment. However, if a gene 1 has 1,000 nucleotides and gene 2 has only 200 nucleotides, then global alignment is not much useful. A general global alignment technique is called as needleman wuntch algorithm and based on dynamic programming. A global alignment between two sequences in an alignment which all the characters in both sequences participate in the alignment. Global alignment are useful mostly for finding closely related sequences. All these sequences are also easily identified by local alignment methods. Global alignment is now somewhat deprecated as a technique. As science just develop many techniques, so nowadays people more focus on local alignment as compared to global alignment. Furthermore, there are several complications to molecular evolution which prevent these methods from being used, because sometimes usually just researchers just try to find out a specific functioning domain or motif, or specific interacting residues of the known gene with the unknown gene, so they just jump into the local alignment as compared to global alignment. Find the global best fit between two sequences. Here we have, you can have a look that here we have a sequence S with these amino acids, and then we have a sequence T with another amino acid, and we are going to align that alignment S and T, V with V, I with I, V with V, A, and here we have a mismatch L with D, A. Then with S we have a deletion here in this sequence, or you can say insertion in this sequence of S. Then we have V with alignment, and then we have two deletions in respect to this sequence. This sequence we have deletion that delete two amino acids. However, if we have a look to this sequence, we can say it is the insertion of two specific amino acid in this sequence. Then we have a mismatch, and then one more alignment. So this is another example of global alignment. So we are going to jump on a look for needleman wuntch algorithm. The needleman wuntch algorithm was produced by was generated in 1970. So here is the article link, General Molecular Biology. You can have a look if you are interested to read that article. The needleman wuntch algorithm performs a global alignment on two sequences, that is S and T, and is applied to align protein or nucleotide sequences. So this algorithm just help researcher to align many proteins globally in early era of bioinformatics. The needleman wuntch algorithm is an example of dynamic programming and is guaranteed to find the alignment with the maximum score. The needleman wuntch algorithm, of course, this work for both DNA sequences as for protein sequences, not for only DNA nucleotide sequences or protein amino acid sequences. You can apply this algorithm on both the sequences. Score of aligned characters are specified by the transition matrix. The similarity of characters as I and J, we will have a look in these matrices, and we will generate a matrix for this one. For example, if the substitution matrix was like this one, here we have a sequence AGCT and then AGCT here also. So here we have some matrix there with the values 10, -1, -3. We will make this a matrix in later slides, then you will get a clear look at how these values come. So then the alignment happens here with a gap penalty of -5 would have the following score, that is S with A and C plus this one, this one. From this alignment, you can see A and C. This is a mismatch. Then G and G match, A and A match. Then there are three. You can see here that A and C, sequence A and C, then G and G, A and A, and then 3 into D, mean 3 gaps. And one gap penalty is equal to -5. Here you can see that A is -3 with C plus 7. Similarly, 10 with then calculate these values total, in total, and this is A, this is C. So you can see that when A and C comes together, value is -3. A and C -3, A is -3. Plus G and G, G and G. When G and G comes, you can see here 7. G and G, 7 is here. Plus 10, 10 with A and A. When A and A comes together, then 10 is here. Similarly, after that, we have 3 with gap penalties, that -1 for one gap penalty, 3 with -15. And similarly, we have this complete value. Suppose here A and G when comes together -1, and here we have -1 here. And when we have S, C and T together, C and T here, we have 0 and we have 0 here. For with the help of this matrix, you can see that we just fill this one with the help of gap penalty, also -5 multiplied by 3 here, and we have a score of 1. So we have to generate the maximum score to align a specific sequence. We will align the sequence later. So if you have some confusion in this, don't worry, we will inshallah solve this confusion in later slides. The substitution matrix, a more realistic scoring function, is given by the biological inspired substitution matrices like point accepted mutation by Dayhoff and Blossom, that is known as block substitution matrix by Hanikoff. So scoring function, the cost for aligning the two sequences. Here we have the two sequences and we align it. You can see that there are 7 matches, 1, 2, 3, 4, 5, 6, 7. We have 1, 2, so 1 and 2 mismatches and 3 gaps. 7 matches minus 2 mismatches minus 3 gaps, then we have 2 in actual as a score for these two sequences. So here is the method we can score any, that is the optimal scoring method in actual. After global alignment, we have local alignment, as we discussed in earlier, that global alignment just have a look to the complete or entire or whole two sequences. However, in local alignment, we just have a look to the patches or motifs or domains. So local alignment method find related regions within sequences. They can consist of a subset of the characters within each sequence. For example, position 20 to 40 of sequence A might be aligned with position 50 to 70 of sequence B. So you can apply the local alignment. This is more flexible technique than global alignment and has the advantage that related regions which appear in a different order in the two protein can be identified as being related. We already discussed in detail that if one protein has 100 amino acids and another protein has 80 amino acids and only a specific chunk or piece of amino acids just have a alignment, so we will jump on local alignment. This is not possible with global alignment method, that to find a specific chunk or portion of the alignment of that specific two proteins. Local alignment is a matching two sequence from regions which have more similar with each other. These are more useful for dissimilar sequences that are suspected to contain regions of similarity or similar sequence motifs within their larger sequence context. Smith and Waterman algorithm is a generally used for local alignment method, also based on dynamic program, as global alignment are also global alignment method is also depends on dynamic programming. Smith and Waterman algorithm was produced in 1981, is for determining similar regions between two nucleotides with the small patches or small chunk. And Smith Waterman is a dynamic programming algorithm similarly to needle-wuntch. And however, it is the improved form of needle-wuntch algorithm and has more specificity to local alignment and to hunt out the specific portion. It has the desirable property that it is granted to find the optimal local alignment with respect to the scoring system being used. The Smith-Waterman algorithm is demanding of time, as it is it has to check all the sequences in patches, and also it needs much memory resource, so it can make so many alignments, and then it can give you a good one. In order to align two sequences of length, m and n depends on the length of two sequences, and also the time and space depends on the two sequences. As a result, it has largely been replaced in practical use by the BLAST algorithm. Nowadays, people just use BLAST algorithm instead of Smith Waterman and wuntch, needleman wuntch. However, the base of alignments are depends on needleman wuntch and Smith Waterman algorithms. Sequence alignment is used to study the evolution of the sequences from a common ancestor, such as protein sequences or DNA sequences. Mismatches in the alignment correspond or gaps or shows the mutations or insertions or deletions of the specific sequence. Sequence alignment also refers to the process of constructing significant alignments in a database of potentially unrelated sequences. So after that, we have a BLAST that is considered as the most fast algorithm nowadays in comparison with Needleman, wuntch, and Smith Waterman. Fast approximate alignment has been performed by BLAST. Fast but heuristic, mostly used algorithm in bioinformatics nowadays. So here you can see this is the BLAST search algorithm, in which if you want to find a query of only 3 words, that is PQG, and neighborhood words, you have this matrix in which you have PQG, PEG, PRG, so many. So this is your query sequence, and this is your subject sequence. You have to check for this one that in this matrix, P is here, G is here. However, here is a gap in place of Q. So in BLAST, it places all the amino acids in between P and G and just give a specific score. Like PQG gives 18, PEG gives 15, PRG gives 14, PKG gives 14. So it gives a big scores. However, here we have PMG. In subject sequence, and in query sequence we have PQG. So in PMG it is the 13th one. Score is 13-1. So it is more than PQN and PQA. So that's why BLAST just jump into the PMG for high scoring segment pair. Depends on high scoring segment pair. Here we have in P space G. So in this one we have PMG. Similarly, we have here PMG. If we have here PHG, then we will jump on 13 also. And if we will have here PQG, then we will jump on 18. However, here was PMG, so 13 was the highest scoring number here. In semi-global alignment, it is a hybrid of global and local alignment. It is also known as global alignment and semi-global alignment. To find the best possible alignment that includes the start and end of one or the other sequence. If you want the end or start of any sequence, suppose query sequence or reference sequence, you have to jump on semi-global. If you want to check end to end from one to the last, you have to jump on global alignment. And if you want to just check the specific portion, chunk, or motives or domains, you have to jump on local alignment. This can be specifically useful when the downstream part of the sequence overlaps with the upstream part of the other sequence. If there are two genes or two proteins, suppose if we will have an example of protein that a protein A has 1 to 100 amino acid, however protein B has 1 to 110 amino acids. However, the portion of protein A from amino acid 80 to 100 is similar to the portion of protein B from 10 to 30, then you have to go for semi-global alignment. Here we have some comparison of global sequence alignment and local sequence alignment. Global sequence alignment made to align the whole sequences. However, local just gives specific regions. Global contains all letters from both the queries and query sequence and target sequence. However, local alignment just have specific portion. It has all the global alignment should have the similar length of both the sequences. However, in local we don't have this type of limitation, suitable for aligning two closely related sequences for global sequence alignment. However, in local sequence alignment, it is suitable for aligning more distantly related sequences. Usually done for comparing homologous genes. However, local sequence alignment used for finding out conserved patterns of DNA, because that conserved pattern has a small number of small number of amino acids as a region. Needleman wuntch algorithm used for global alignment, and Smith Waterman used for local alignment. Examples for global sequence alignment are embossed, needleman, wuntch, blast, and these. However, for local sequence alignment, we have an example of blast, emboss, water, and L-align. After that, we have multiple sequence alignment. That is also a good concept for multiple sequence alignment. For multiple sequence alignment, we have to align more than one sequences at a time and check the conserved portion from all of these sequences, no matter it is of protein sequences or DNA sequences. Multiple sequence alignment is an extension of pairwise alignment to incorporate more than two sequences into an alignment. Multiple sequence alignment methods try to align all of the sequences in specified set, and the most important tools, or the most familiar tool nowadays, is cluster. That was previously known as cluster W. However, nowadays it is named as cluster omega for multiple sequence alignment. Here we have an example of four different sequences that that align with the help of multiple sequence alignment. You can see that from 1 to almost 579 amino acids present in these sequences. So these three are the sequences from different organisms, you can say, and this is the consensus. That just showed, the statics shows that the gaps, and then this K shows in red color that all this region is conserved in all these. However, the blue color shows that it is the most similar one, like in 2 mm, and this one has some mismatch. So red color explains that this region is conserved in all three sequences. However, blue color show that it has two sequences has that similar amino acid. However, the statics shows gaps, that here it is gap in all of the not gap, mismatch, in actual mismatch, like F with Y with R. Similarly, here we have P with E with M, and also represents gaps here, like no other amino acid present with these two sequences for this one. So with this steric just shows gaps and mismatch, and the red color shows similar ones, and the blue color shows more than two sequences has the similar property. So this is the multiple sequence alignment. If you just open cluster omega on your Google and just write cluster omega, just give 3 or 4 sequences and you can have the look to all the sequences. So we discussed about dynamic programming in our previous slides, and we just said that global sequence alignment and local sequence alignment has performed by dynamic programming approach. So the question is, what is dynamic programming approach for sequence alignment? The dynamic programming approach to sequence alignment always tries to follow the best prior result so far to give the most accurate result. It is not in actual the programming as we discussed in our fundamental of programmings or programming languages of computers. In actual, it is the set of rules that just apply to solve the specific problem. Try to align two sequences by inserting some gaps at different locations so as we can get the maximum score of the alignment. Try to align the sequence and give some insertions and deletions and gaps for having a maximum scoring alignment. Scoring measurement is determined by match award, scoring measurement depends on three different types of values. That is, the first one is the match award, that if there is a match between two amino acids or two nucleotides, the specific number has to give. After that, there is a mismatch as a penalty, and gap penalty that also has some negative or zero. Specific score gives with the help of score measurement. I'm sorry. Back to the point. So we were discussing about the score measurement that with the score we have to give a specific score when there is a match between two amino acids or alignment or two nucleotides. There will be a specific score. Suppose for match we will give one score. For mismatch we will give -1, like with A with 3, and with gap penalty if we have to place a gap there, then we will give a score of -2. The higher score, the better alignment we will have. If both penalties are set to 0, like the penalty of mismatch and penalty of gap will set to 0, it aims to always find an alignment with maximum matches so far. Maximum match is equal to largest number matches can have for one sequence by allowing all possible deletion of another sequence. It is used to compare the similarity between two sequences of DNA or protein to predict similarity of their functionality. So dynamic programming is used widely in bioinformatics for the task of aligning different sequences of protein or RNA or DNA. Needleman and once describes general algorithm for sequence alignment, maximize a score of similarity to give maximum match. Maximum score, as we already discussed, that the largest number of nucleotides that can be matched with others will give the maximum score. As you know that we discussed that the match will have a positive score, however gap penalty or mismatch penalty will have a negative score. So when we have maximum matches, then we will have maximum positive scores, and maximum positive scores will always be in larger number as compared to negative scores. That's the inactual concept of the aligning of two sequences. So it was introduced, the dynamic programming method was introduced by Richard Bellman in 1940. The word programming here denotes finding an acceptable plan of action, not computing programming. As we already discussed, that it is the set of series that helps to solve the alignment problem. It is not the specific programming of a computer language. It is useful in aligning nucleotide sequences of DNA, amino acids, and proteins. Dynamic programming method helps to solve complex problems by breaking them into simpler problems. Problem can be divided into many smaller parts. Polyas, Polya. There was a concept by Polya's concept. Polyas also give four different steps to solve a problem for problem solving in computer sciences. If you have recall that, first one is to understand the problem, second one is to deceive a plan, third one is to implement the plan, and fourth one is to evaluate the plan. This is also similar like that. Dynamic programming is a three-step process that involves initialization, matrix filling, and traceback and aligning. First of all, we have to initialize the alignment, then we have to fill a matrix, then we have to give a score on the base of penalties and matches and mismatches, and then we just trace back that that score to align to have the alignment. In initialization, the first step is the global alignment dynamic programming approach is to create a matrix with M1 columns and N1 rows, where M and N correspond to the size of the alignment. Matrix filling, we have to fill the matrix with penalties in the form of diagonal or from upside to down or from horizontal or vertical. And then we will move back to the trace backing. In initialization, first of all, we have to generate a matrix, and matrix should have all the values in rows and columns. However, in matrix filling, we have to fill the matrix with the given score of matches, mismatches, penalty, and gap penalties. And these values just move downward through diagonal shape or horizontal shape or vertical shape. And then we have traceback and alignment concept. In global alignment via dynamic programming, first of all, we have to fill all the empty rows and columns and fill the first block with 0. There are some set of rules that we have to follow for performing that global alignment via dynamic programming. In this section, this is very important section, so everyone should have to be very focused and try to understand this alignment, how we are going to do the global alignment. Now we are going to perform the global alignment to understand the concept basically. First of all, we have two sequences, sequence A and sequence B, suppose ATGC and ACGT. and we want to align these two sequences. And how we will get the highest global alignment score? First of all, we have to fill all the rows and columns of this matrix, in which first block we should have to fill with 0, and after that we have to fill all the columns and rows with different penalties. While filling the matrix, there are three possible values: from horizontal, or from horizontal score, from vertical score, from gap penalties, from mismatches. Here we have a vertical score plus gap penalty, and in if we just move from vertical direction or in horizontal direction, we have to plus the gap score with the given score. However, if there is a match or a mismatch, then we have to move diagonal. We will have a look in next slide in detail about that. If we will give the three different scores, like if there is a match between A and A, then we will give +1. If there is a mismatch like A and T, then we will give -1. And when there is a gap penalty, we will give the score -2. So you guys have to memorize some concepts. First one, we have to fill all the rows and columns. Second one, we have to give 0 to the first block. Third one, we have to fill all the metrics with the help of the scoring. If there is a gap penalty, we will move vertically or horizontally. However, if there is a match or mismatch, then we will move diagonally. And these score, just keep it in your mind. When we have a match, we will write +1. When there is a mismatch, we will write -1. And then when there is a gap penalty, we will add -2. So here we have a matrix. Be very focused. Here we have a sequence AAAC. We just write it here, AAAC. Then we have a sequence AGC, AGC. Don't worry. First, we will try to understand it very well on this matrix and other matrix of local alignment. After that, I will solve it on a paper, and I will show you how we just calculate this one more time, so we can understand it very easily. It's not difficult. It's very easy. You just have to be focused. Don't be panic. Don't be consider it very hard. Just take it very light. Just focus. Here we have a sequence AAAC, AAAC. Then we have a sequence AGC, AGC, and we have to align these two sequences. As we know from the rules from this slide, that from the first block we have to write 0. So we write it 0 here. After that, we just said that for gap penalty, we will have to add -2 into the previous value, that is horizontally or vertically. So always you have to check it like this way: A with A, A with A, A with A, A with C, then A with G, A with G, A with G, C with G. Similarly, A with C, A with C, A with C, and C with C. So we have to fill the column like this way, all the columns and rows. Similarly, we just place it 0 here. Now we have to check with A with this gap. So here is a gap. And when we have a gap, we have to add -2. So 0 plus -2 -2. Then A with this gap. Now we have to add -2 in this value, the previous one, because we are moving horizontally. -4. Now A with this gap, -4 -2 -6. C with this gap, -2 -6 -8. Similarly, A with this gap, A, G with this gap, C with this gap, we add -2 every time. So we have these columns. Once again, we have this sequence AAAC, AGC. We place it 0 here as per rule. Now we have to fill the matrix. If there is a gap, we have to add -2 in the previous value. So with this 0, we have to add -2. Then -2, we have to add -2, and then it is -4. For this -4, we have to -2 again as a gap, -6, -8, and similarly, we fill this column also. After that, we are going to check this A with this A. And we discussed that if there is a match, we will take the diagonal score. And if there is a gap penalty, we will take the score from vertically and horizontally. So if we have a match like A and A, we will add 1. That was we describe here. If there is a match, we will +1. So we will +1 into this 0 and add here. 0 plus 1, 1. And when there is a gap, -2, -2, -4, -2, -2, -4. So we have three values in this box: -2, -2, -4 from this side, from this side, and 0 plus 1, 1 from this side. So we have to take the maximum score and has to write here. So -4, -4, 1, 1 is the maximum one. So we write it here in this column. Similarly, after that, we have A with this A. And again, we have 1 for positive for match and -2 for gap penalty. -2 and -4, -6, 1 and -2, -1, -2 and -2 and +1, -1. So we again have -1 here from this side and this side. So we have to make an arrow. Similarly, we just fill all these boxes. I will do it on a paper for you so you can understand more clearly about that. So don't worry about that. After that, we have to move from the maximum value to trace back to have that specific sequence. In backward tracking, we have to move from last cell that has on the lower corner, like here, from this cell. And remember, the last, these 4, 5 values will have the maximum value, maximum number. Like from -4, -2, -1, -1, -1 is the biggest value. Similarly, when you will be in the last, you have the maximum value. So now we have to align this sequence backward with the help of these alignment. Local alignment via dynamic programming, similarly, like global alignment, first we have two sequences. In that one, global alignment, we just place a 0 into the first box. However, in local alignment, we will fill all the first row and first column with zeros. This is some exception in local alignment as compared to global alignment. If the values come in negative, in global alignment, we just add penalty as -1 mismatch and gap for -2. However, if we have minus value in local alignment, we just place 0 at that point. Here you can see with match we have 1, with mismatch we have 0, with gap penalty we have 0. Similarly, we have this matrix. We have a sequence GAATTCAGTTA, sequence GGATCGA. So first we write this sequence here, then sequence here. As with the rule, we just place 0 on this position, this row, first one, and 0 on this column. Once again, if we have a match like G and G, we have a match and we have one score, we will plus in this one, and gap penalty 0 and 0. When we have 0, 0, and 1, we will add the first maximum value, that is 1. So with the help of this one, we just generate that matrix, and we have to make the row arrows so we can backtrace is easily. Don't worry, I will make a matrix on paper and upload for you so you can understand it very easily. And then we have a backtracing, how to just generate the alignment with the help of that specific arrows we made on the matrix. Now I am going to generate the matrix on paper, and I will upload that for you too. So don't worry, just try to understand, focus. It's not much difficult, but it is the base of bioinformatics, global alignment and local alignment. We have a quick look to the local and global alignment and the basic concepts, the principles. Now we are going to perform how these alignments works. I will try to take the same matrices I used in this example on paper, and then I will try to use another one example so we can understand it very easily. Thank you so much everyone for joining.